Welcome to Lecture 11.2, in which we will go over DNA replication and repair, specifically of eukaryotes. This is Dr. Matthew Meselson, who along with Dr. Franklin Stahl, established that DNA replicates in a semi-conservative mechanism. This is Dr. Tsuneko Okazaki, who with her husband, Dr. Reiji Okazaki, discovered Okazaki fragments created during DNA replication. Dr. Elizabeth Blackburn, along with Dr. Carol Greider, discovered the enzyme telomerase, which is used to extend telomeres. We will talk about what each of these discoveries are throughout this video. Our learning objectives for this video are to describe the process of DNA replication and the roles of the enzymes involved, understand what telomeres are and how they protect DNA, describe how DNA polymerase can perform proofreading during DNA replication to reduce error rates, and describe different mechanisms of DNA repair after DNA has been replicated. As discussed in the previous video, the two strands that make up DNA are complementary, which means that if we know the sequence of one strand, we also know the sequence for the other strand. This nature of DNA allows for easy replication of DNA, and it's part of why it's so effective for the storage of genetic information. It was discovered that DNA replicates with a semi-conservative mechanism. This means that when DNA is replicated, we start off with two original strands of DNA that separate, and a new complementary strand is created based on the old strands. The result is two double-stranded DNA, in which one strand is the original template, and the other half is the newly synthesized strand of DNA. The complementary strands are always synthesized in the 5' to 3' direction. Since the strands are anti-parallel, that means that synthesis of a new strand of DNA starts at the 3' end of the original template DNA, which is the 5' end of the new DNA. There are three stages of eukaryotic DNA replication: initiation, elongation, and termination. We will see that these are also the names of the three general stages of transcription and translation later on. In replication, initiation occurs when the DNA is separated and made accessible to proteins and enzymes involved in the replication process. When we learned about how DNA is replicated in prokaryotes, we learned that it begins at an area called the origin of replication. It's the same for eukaryotes, except there are multiple origins of replication along the linear strand of DNA. The enzyme helicase is responsible for unwinding the double helix and opening up the DNA. Think helix, helicase. As a helicase unwinds the DNA, Y-shaped structures are formed. These are called the replication forks, and there are two at each end of the origin of replication. Like in prokaryotes, replication occurs bidirectionally. Elongation is a step in which nucleotides are added to form a DNA strand, and therefore, the step in which the DNA strand is elongating. However, DNA can only be added if there is a 3' hydroxyl group available to connect to the phosphate on the 5' end. RNA, on the other hand, does not have this problem. Remember that RNA is very similar to DNA, but it has a second hydroxyl group on its second carbon. An enzyme called primase puts down primers made of RNA. This RNA is complementary to the template DNA and creates a 3' end that DNA nucleotides can be added onto. In other words, it essentially primes or prepares the DNA for elongation. The primers are later removed and replaced with DNA so that there are no RNA-DNA hybrids. The enzyme that is actually responsible for elongating the DNA is called DNA polymerase. It adds nucleotides to the 3' end of the DNA, and therefore works in the 5' to 3' direction. You can think of it as the enzyme that is taking nucleotide monomers and putting them together to form a polymer, hence polymerase. Again, elongation continues bidirectionally. It also continues on both strands. The leading strand is a new strand that is made continuously. Looking at the diagram, this top strand of DNA is being opened up in the 3' to 5' direction. Since DNA is made in the 5' to 3' direction, and the new strand's 5' end is where the 3' end of the template is, that means that the DNA polymerase is able to smoothly make DNA while trailing behind the helicase. On the other hand, the bottom strand is being opened up from the 5' to 3' direction. However, the new strand still needs to be made in the 5' to 3' direction. This means that the new strand needs to wait for the helicase to open up the DNA, then put primers, and then synthesize DNA in short stretches, stopping when it reaches the beginning of the primer that was previously set down. Since the strand has to wait for the DNA to further open up before it can continue synthesis, it is called the lagging strand. These short stretches of DNA are called Okazaki fragments. Termination of replication in eukaryotes is not very well understood. However, it is thought that it occurs when two replication forks meet one another and converge. During this process, the RNA primer is removed and replaced with DNA. There are also gaps between the Okazaki fragments since the nucleotides that are next to each other are not actually bonded to one another. An enzyme called DNA ligase performs ligation, which really just means that it seals or glues two nucleotides together. It essentially takes the 3' end of one nucleotide and attaches it to another existing fragment of DNA. One of the problems with DNA replication of linear chromosomes is that the ends of the chromosomes also need to be replicated. These two strands represent the original strands of DNA, with the end of a chromosome on the right. The strands separate, and new strands are synthesized. The lagging strand has to set down a primer at the end of the chromosome, while the leading strand can easily replicate the end of the chromosome. After the primers are removed, all of them will be replaced with DNA, with one exception. Since there is no longer a 3' end to add nucleotides to, the primer set down at the very end cannot be replaced. As a result, the lagging strand will always be unable to replicate the very end of a chromosome. Of course, it would be bad if we lost some of our genes every time our DNA is replicated. This is where telomeres come in. Telomeres are repetitive sequences found at the ends of linear chromosomes. Importantly, they do not code for any genes, so losing some of these sequences does not affect our genes at all. Their role is to protect the ends of the chromosome. They do become shorter with each round of replication, and it's thought that cells can replicate about 50 times on average before its telomeres run out. Some cells have telomerase, which is an enzyme that lengthens the telomere so that the cell can continue to divide. However, most adult cells no longer have telomerase. That's why there is an association between the length of telomeres and age. Telomeres would be shorter in people who are older. There is also a relationship between telomeres and cancer. Cancer cells sometimes start expressing telomerase again so that they can lengthen their telomeres, allowing them to replicate an unlimited number of times and form a tumor. During replication, the DNA polymerase can make mistakes. If these mistakes aren't caught, this can lead to a mutation, which is a permanent change in the DNA sequence. Mutations can be beneficial, and we will later learn how they are the main driver of evolution. However, they can also be harmful, especially if the mutation is in a gene that codes for a protein. Luckily, the polymerase is able to check its mistakes. Incorrect bases can generally be recognized because they will not pair properly with the base on the original strand, creating a bulge. The DNA polymerase has its own proofreading mechanism in which it can recognize the mistake, then act as an exonuclease to remove a nucleic acid that it just placed at the end of the strand. It can then add the correct base in its place. Of course, mistakes can still happen even with proofreading. As its name suggests, mismatch repair is used to correct mismatched base pairs that were not caught by the polymerase during replication. Instead of an exonuclease, an endonuclease is used to cut the nucleic acids in the middle of the DNA. This removes a segment that is filled in by a polymerase, and ligase is used to attach the nucleotides together. DNA can also get damaged, and the damage can also lead to mutations. One of the mechanisms for repairing damaged DNA is base excision repair. This repairs damaged single nucleotides. For example, say this G in red is a damaged G. It will get recognized, and at first just the base will be removed or excised. Then the rest of the nucleotide will be removed, and the correct nucleotide will be put in its place. Nucleotide excision repair is used for slightly bigger damage that distorts the double helix, such as two thymines next to each other that get bonded by UV light, creating thymine dimers. This type of damage is repaired by removing a chunk of the DNA that contains the damage, then filling the chunk back in using DNA polymerase and ligase. In this video, we learned that DNA replication begins with helicases unwinding the DNA double helix. Primase is then used to set down RNA primers that allow for elongation to begin. DNA polymerase is used to synthesize DNA in a 5' to 3' direction during elongation. The leading strand is made continuously because the DNA polymerase that makes it moves in the same direction as a helicase, while the lagging strand has to be made in fragments because it moves in the opposite direction of the helicase and needs to wait for the helicase to open the DNA up more before it can keep synthesizing. During termination, the RNA primers are removed and replaced with DNA, and the enzyme ligase is used to seal the gaps between nucleotides, joining the hydroxyl group on the 3' end of one nucleotide to the phosphate group on the 5' end of the other. Since the end of the lagging strand can't be completely replicated, there are telomeres present at the end of the DNA to protect it. During replication, the DNA polymerase has its own proofreading activity that allows it to catch errors. Errors that are not caught are fixed by mismatch repair, while damaged DNA can be fixed by base excision repair or nucleotide excision repair. In the next video, we will begin talking about RNA and transcription. Thanks for watching.